Hello students, welcome to EPG Pathshala. I am Monica Saini from Department of Anthropology, University of Delhi. Today I am going to talk about the module Differentiation of Animal and Human Hair under the Forensic Anthropology paper. Through this module, you will be able to differentiate between human hair and animal hair, and you will also be able to know the general structure and different types of humans and animal hair. Presence of hair on skin is unique feature of human beings. Each hair consists of the part within the follicles, the hair root and shaft projecting the body surface. The hair is nourished by a hair follicle. Hair follicle produce keratinized cell which push the shaft from below and spread the oily secretion over the shaft. The entire hair consists of three regions: medulla in the middle, a thicker cortex around the medulla, and a delicate covering of cuticle which is made up of keratinous cells. Hairs are differentiated with their characteristic length, color, shape and root appearance. Hair is present on many different regions of the body, each region such as the head, pubic area, chest, axillae and limbs. These all parts have hairs with some microscopical characteristics attributable to that particular region. The morphological examination routinely involves a two-step process involving a comparison microscope. The first step is the identification of hairs and the second step is the comparison of questioned and known hairs. During comparison, the length of the hairs as well as the maximum number of microscopic features is considered. Presently, it is a widely accepted tool to identify the age, sex, color, race, disease profile, diet, occupational and environmental exposure, metal poisoning, geographical indicator, illegal wildlife trade, sexual assault, rape, disputed maternity and paternity matters, and in cases of mitochondrial DNA examination, where the questions are raised related to the evolution and inheritance. Hair is always considered to be the associative evidence and its ultimate objective being to associate a suspect and the victim in the act of crime supported by the evidences based on the scientific approach and the crime scene scenario. Hair is a fiber, thin structure approximately around 0.1 millimeter in diameter. It has an oval or a circular cross section. Hair is mainly composed of three concentric regions, that is, the outer layer being the cuticle, which is a thin coating covered by the tital scales. The main constituent being cortex, contributing to almost 90% of the total weight of the hair, made up of differently shaped cells specific to the hair type, the keratin. Fibers are arranged in a honeycombed arrangement of cell structure containing air pockets continuously or discontinuously called the medulla. The follicle is the most active part which lies beneath the skin surface producing keratin protein, and the hair shaft collectively make up the hair strand. The follicle associated with the hair shaft acts as a mold to shape the strand. A hair grows from the papilla, and with the exception of that point of generation, it is made up of dead cornified cells. It consists of a shaft that projects above the skin and a root that is embedded in the skin. These elements are deposited in the hair during its growth and absorbed by the hair from the external environment. The hair shaft is stiff to a greater extent that helps reduce damage caused due to the shear and stress in the strand. The frictional behavior is mainly linked to the cuticle, which along with the cortex also determines the tensile strength and smoothness of hair condensation of alpha amino acid in the hair that are bound together in the same plane. Hair fiber is not uniform in composition, and it is made up of both crystalline and amorphous regions. The segment of hair that captures the X-ray photograph of the keratin content is found to be around 20% and 80% crystalline and amorphous respectively in nature. The keratin fiber is chemically composed of two forms, that is alpha keratin and beta keratin. The alpha form tends to remain folded, but when it is stretched, it corresponds to the beta keratin form. So students, now I will tell you about the different types of human hairs. Head and body hair of humans is classified as intermediate hair, combining the characteristics of bristle and wool hairs. 4 types of hair appear on the bodies of humans. The first type is primordial hairs. These hairs appear as early as the 3rd month of gestation, growing on the upper lip, the eyebrows, the palms, and soles of the fetus. These hairs gradually disappear and are replaced by the softer lanugo hair over the entire body. The second type of human hair is lanugo hair. Lanugo hairs are normally shed after the 6th month of gestation. They are fine, soft, unmodulated, and normally unpigmented hairs. The surface of lanugo hair is smooth with almost indiscriminate scales. It is replaced by the vellus and terminal hairs. Lanugo hairs is often observed on an absorbed fetus, and these can be useful in investigation of possible infanticide. The third type of human hairs are vellus hairs. The vellus hairs are the fine, soft, unmodulated hairs spread uniformly over the body surface. They rarely are more than 2 cm in length. The last type of hair is terminal hair. Terminal hair is found on the scalp, eyebrows, eyelashes, and to a lesser extent, the limb of both sexes. Puberty is accompanied by pubic and axillary hair growth such as hair of the face, chest, back, arms, and legs. The various terminal hair types can be distinguished by density and morphology. Next, we will study the type of hairs in animals. There are basically 3 types of hairs usually found in animals. The first type is the vibrissa. These hairs are the whiskers of many animals. They are normally tactile and sensitive, such as the whiskers on a cat. The second type of hair is bristle. This is the coarse bristle that provides an animal with a protective coat. These guard hairs can readily be identified by their distinctive appearance and morphology between various animal families. The third type of hair, which is usually found in the animals, is wool. Wool or fur, they provide insulation from the wet and cold environment. These fine hairs cover the bodies of all mammals. According to the Dedrick and Koch, animal hair can be classified into three major groups on the basis of their microscopic appearance. These types are deer family and antelope hair, commercial fur animal hair, and the third is the domestic animals hair. For individual identification in these groups, deer family and antelope hairs are distinguished on the basis of their scale patterns, whereas the commercial fur animals are distinguished on the basis of their color bands, scale patterns, and medullary structure. Domestic animals are distinguished primarily through their root structure, medullary structure, and pigmentations. Other type of hairs found on animals include tail hair and mane hair. Human hair is not so differentiated and might be described as a modified combination of the characteristic of guard hair and fur hairs. So students, there are three types of animal hairs. First, we will talk about the hairs of deer family and antelope. The hairs of the deer family and antelope are very coarse and the overall diameter is approximately 300 micron. Medulla is composed of spherical cells that occupy the entire hair. The diameter of the hairs of deer family is constant throughout, and most of the hair, this diameter part is constant. The root of the hair is wine glass shaped and regular wave or crimp in the hair is generally found. Whereas the commercial fur animals, they include several specimens commonly encountered in the fur garments that are distinctive in their microscopic appearance. These are hairs of rabbit, seal, mink, muscat, and chinchilla. Of these hairs, seal and chinchilla are identified on the basis of their appearance of their down or fur hairs, but not by their guard hairs. Commercial fur animals, their hairs are very fine to medium and the overall diameter is 20 to 150 micron. The characteristic medullary formations are serial or vacuolated. There is wide diameter variations are also seen in single hair and hairs are generally banded. So, there are wide variation in the color and length of the hair specimen in the domestic animal group, which makes up the third category of animal hair. The identifying characteristics are generally, and they are basically applied in most cases. The domestic animal hairs have medium to overall diameter, that is 75 to 150 micron. The medulla is generally amorphous and the moderate diameter variation in single hair is seen, and the hairs are generally unbended. Now we will see the pigmentation of animal hair fiber. Melanin is mainly responsible for many shades of black, brown and other shades of hair, and is found in the form of granules which are seldom uniformly distributed in fiber, or in crimpy fibers the pigmentation is asymmetrical. More pigmentation is observed in paracortex than orthocortex. This asymmetrical distribution of granules is originated by differential allocation of melanocytes inside the follicular bulb where the fiber is formed. Electron microscopy of the melanin granules revealed their egg-shaped bodies. The ratio varies in major and minor axis with the origin of granules. The pigments get deposited over the layers of the granules in hair made up of layers formed of proteins. Electron micrographs showed the axis of granules tends to lie parallel to the axis of cortical cells, and the granules are arranged end to end on rows covered by fibular keratin. Melanin is chemically inert in nature and are polymers of high molecular weight and known to be formed due to the action of tyrosinase to yield indole 5-60-quinone, which then polymerizes. Several researches have revealed that natural melanin show close association with metals, especially iron, and with proteins. Thus, pigmentation of hair can serve as important parameter for differentiation of animal hair from human hair. Differentiation in animal and human hair. The human hairs are generally consistent in color and pigmentation throughout the length of the hair shaft, whereas animal hairs may exhibit radical color changes in a short distance called bending. The distribution and density of pigment in animal hairs can also be identifiable features. The pigmentation of human hairs is evenly distributed or slightly more dense toward the cuticle, whereas the pigmentation of animal hairs is more centrally distributed, although it is more dense toward the medulla. The medulla, when present in human hairs, is amorphous in appearance, and the width is generally less than one-third the overall diameter of the hair shaft. The medulla in animal hairs is normally continuous and structured, and it generally occupies an area of greater than 1/3 the overall diameter of the hair shaft. The root of human hairs is commonly club-shaped, whereas the root of animal hairs are highly variable between animals. The scale pattern of the cuticle in human hair is routinely imbricate. Animal hairs exhibit more variable scale pattern. The shape of the hair shaft is also more variable in animal hairs. The display table is showing you the main differences in human and animal hair on the basis of pigmentation, medulla, and scale structure. The pigments in human hairs remain constant in its color, and pigmentation may be present throughout the length of the shaft, whereas in the animal hair, the pigmentation shows rapid and radical color changes, which is also referred as bending. Medulla in human hair is thin, taking up no more than 1/3 of the hair shaft diameter, and this medulla is also amorphous in the structure. When we talk about the medulla of animal hair, then it is very wide and its structure is very regular and well defined with certain patterns. Scale structure in the human hair, it has overlapping imbricate or flattened scales with narrow margins. In animals, the scale structures have coronal or crown-shaped scales, or it may be spinous or petal-shaped scales. The presented diagram is showing you the differentiation in the human and animal hair on the basis of medullary index. Medullary index is the ratio of medullary width to the hair width. In the first diagram, the illustration of cattle hair is shown where the medullary index is 0.50 or more, whereas in humans the medullary index is 0.33 or less. In the humans, the medulla structure is often fragmentary or trace, discontinuous, broken, or it may be continuous. So students, let us see what we have learned from this module. Hairs are biological specimens and are subject to variation. During the analysis of hair, the examiner must establish the range of variation in the sample and then determine whether the questioned hair fits in that range. The possibility cannot be dismissed that there may be two hair samples whose range of variation overlap, and distinguishing process between the samples is not possible. Difference can be drawn through the consistency in the color and pigmentation throughout the length of the hair shaft. The distribution and density of the pigment in animal hairs can also be used as one of the important identifiable features. Animal hairs exhibit radical color change in a short distance called bending. The pigmentation of human hair is evenly distributed or slightly more dense towards the cuticle, whereas the pigmentation of animal hairs is more centrally distributed, although more dense towards the medulla part. The medulla, when present in the human hairs, is amorphous in the appearance and the width is generally less than 1/3 of the overall diameter of the hair shaft. The medulla in the animal hair is normally continuous and structured, and it generally occupies an area of greater than 1/3 of the overall diameter of the hair shaft. The root of the human hair is commonly club-shaped, whereas the root of the animal hairs is highly variable between animals. The scale pattern of the cuticle in human hairs is routinely imbricate. Animal hairs exhibit more variable scale patterns. The shape of the hair shaft is also more variables in animal hairs. Thank you.